Hi everyone, my name is Dr. Margaux Kenwood. I'm a postdoctoral researcher at the Department of Psychiatry at the University of Wisconsin Madison in the US. I'm also the 2021-2022 Brad Fellow, and I'm excited to share with you all my research concerning the neurobiological substrates of anxiety disorders, which will hopefully pave the way for better interventions aimed at alleviating suffering associated with those disorders. Our work is focused on understanding how individual differences in brain structure, function, and composition are related to individual differences in anxiety. And so what we hope to do with this information is use it in order to come up with better treatments, since we know that a lot of people that are suffering from anxiety disorders are not really getting the treatments that they need. We use animal models in this particular question, and that's because we have a lot of methods that are available to us only in animals that allow us to really selectively target different cell populations, different molecules, modulate their activity, and understand how that interacts with the individual differences in anxiety that we characterize. Rhesus monkeys have a lot to tell us about human pathological anxiety. Like humans, rhesus monkeys have naturally occurring individual differences in anxiety, and we're interested in understanding the brain systems and the cells and molecules that are involved in mediating those individual differences with the hopes of better understanding human pathological anxiety. Whenever we write a grant, we always think about what's the most appropriate model organism in which to perform these particular set of studies. It's actually built into the grant writing process. You do have to justify your choice of model organism. We have a lot of practices in place to ensure the welfare and safety of animals that we use in a research setting. A lot of these are enforced by various regulatory agencies, both at an institutional level, at the level of the animal care facility itself, and then at the level of the federal granting agency that we receive money from. Yeah, I get this question a lot. I think that for some of our studies, there are elements of it that can be better moved to a dish. For example, we're interested in characterizing the way that cells respond to certain pharmacological agents, and for that, we've started moving towards cell culture in order to kind of parse those mechanisms. That being said, a lot of our research does rely on complex interactions between brain regions, which we haven't really developed the methods to approximate in a dish yet. So maybe in the future, yes, but I think that's kind of a distant future in the way that I see it right now. Part of my commitment to communicating about animal welfare and the importance of animal research is being the 2021-2022 Brad Fellow. Brad is Biomedical Research Awareness Day. It's an event that we put on yearly where we send you a variety of tools that you can use to set up an event where you communicate to your peers or to the public about the vital role that animals play in biomedical research. And so if you're interested, come check us out.